Hi, my name is Kristina and I'm a master's student at Université de Sherbrooke in Quebec, Canada. Today I'll be presenting my project on the analysis of a snoRNA-RBP interactome, which led to a discovery of a potential snoRNA-guided splicing regulatory complex associated with a U5 snRNP. So first of all, what is a boxidy snoRNA? It is a type of non-coding RNA known for guiding 2' ribose methylation during ribosome biogenesis. To perform this function, it forms a snoRNP with its core RBP interactors. Recent studies have suggested other roles for boxidy snoRNAs, including the regulation of alternative splicing. And some of these roles are thought to be mediated by interacting with RBPs that were not previously characterized as core snoRNA binders. These new discoveries led to our hypothesis that snoRNAs can have a wide range of functional roles in gene regulation by interacting with various RBPs, overall having a combined regulatory effect on protein-coding target RNAs. So to study the extent of these interactions and understand their functional consequences, we built the snoRNA-RBP interactome. The snoRNA-RBP interactome includes 3 different types of interactions. First, the RBP binding to a snoRNA. Second, the 2 RBPs physically binding to each other. And lastly, the overlap of targets between a snoRNA and an RBP. And here I would like to emphasize that it is not just an overlap of targets by random chance, but we searched for snoRNA-RBP pairs that had enrichment of overlapping binding sites on the same target RNA. We focused on these 3 interaction types because eventually we are interested in a snoRNA-RBP pair that forms a complex to have a combined regulatory effect on its shared target RNA. And these potential snoRNA-RBP complexes can be explored by searching for snoRNA-RBP pairs that share interaction types number 1 and number 3. The snoRNA and RBP interaction data were obtained from various databases, both experimental and computational. These interaction datasets served as a basis to compute the 3 interaction types mentioned on the previous slide. The snoRNAs and RBPs to be included in the network were chosen based on their expression level, and additionally the number of copies for snoRNAs, as many of them exist in multiple copies due to cis recombination. Earlier I mentioned that one of the newly discovered roles for boxidy snoRNAs include the regulation of alternative splicing, but only few such examples have been validated and their molecular mechanisms are still unknown. So we decided to narrow down our focus to the interactions between snoRNAs and splicing factors and look at their functional consequences in gene regulation. So after compiling all the interaction data, we obtained a splicing-specific snoRNA-RBP interaction network. And from now on, we'll be focusing on SNOR22 to look at its interactors and its potential role in splicing. If we go back to our model of interest, I mentioned that this model could be achieved by looking at snoRNA-RBP pairs that share interaction types number 1 and number 3. And in fact, SNOR22 exhibits these interaction types with 2 different RBPs, EFTUD2 and PRPF8. If we look at the subnetwork composed of SNOR22, EFTUD2, and PRPF8, we can see the presence of all 3 interaction types, as well as quite a lot of target RNAs that are bound by SNOR22 and at least 1 RBP at the same site. So what would this snoRNA-RBP complex look like, and how would it interact with its targets? Based on the predicted structure of SNOR22, the main interaction region used by SNOR22 and the binding sites of the 2 RBPs on SNOR22 were identified on the opposite sides of the SNOR22 structure. Additionally, we looked at the binding profiles of these 3 molecules. What's interesting is that SNOR22 alone shows enrichment of binding near the 3' splice site, which is often alternative. On the other hand, the 2 RBPs show enrichment of binding near the 5' splice sites. However, if we focus on the overlapping binding interactions between SNOR22 and at least 1 RBP, we start to see enrichment of binding at both 3' and 5' splice sites, as well as enriched 3' splice site motifs, which partially resembles the binding profile characteristics of SNOR22. In fact, it turns out that EFTUD2 and PRPF8 are core proteins of the U5 snRNP, which is a critical component for pre-mRNA splicing. And the U5 snRNP interacts with both the 5' and 3' splice sites of its targets, bringing the 2 exon ends together. So based on these findings, our hypothesis is that SNOR22 recruits EFTUD2 and PRPF8 to the 3' splice site, of specific splicing events, and they collaboratively regulate the splicing of these shared targets. To validate the functional effect of the snoR22 EFTU-D2 PRPF8 complex on splicing regulation, we are currently doing knockdown experiments for each molecule, followed by RNA-seq and PCR analyses. These are preliminary results, but overall, our knockdowns were quite successful. For snoR22, we were able to follow up with RNA-seq analysis and identified numerous genes that are alternatively spliced between the control and the knockdown condition. To give you an example, here's a cassette exon of USP36, where its splicing events may be potentially regulated by the snoR22 EFTU-D2 PRPF8 complex. The RNA-seq results show exon inclusion only in the presence of snoR22, which hints the possibility of snoR22 being involved in the regulation of this splicing event. We can also see that snoR22, EFTU-D2, and PRPF8 all bind at the 3' splice site, as well as EFTU-D2 and PRPF8 binding at the 5' splice site. A similar pattern is observed from the PCR analysis. When snoR22 is knocked down, the psi values drop by more than 20%, again indicating that the presence of snoR22 promotes the inclusion of the cassette exon. We are currently working on RNA-seq and PCR analyses for the two RBPs, and if these results resemble those of snoR22, then this further supports our hypothesis that snoR22 recruits EFTU-D2 and PRPF8 to the 3' splice site of specific splicing events, forming a complex to regulate the splicing of these shared targets. To conclude, we built a snoRNA-RBP interactome to explore potential snoRNA-RBP complexes involved in the regulation of splicing, and we introduced the snoR22 EFTU-D2 PRPF8 complex as a potential splicing regulator. In terms of the next steps of this project, we plan to experimentally validate the snoRNA-RBP interactions that compose the complex, as well as further investigate how the snoR22 EFTU-D2 PRPF8 complex regulates alternative splicing events. So yeah, that was a quick summary of my project, and if you have any questions, please stop by poster B9 during RSG poster session 2 or leave a comment below. See you soon!